So monosaccharides can form hydrogen bonds with hydroxyl groups, and that gives them high melting point, and they are solid and soluble in water. So because of those hydrogen bonding taking place, those are strong interactions. So that's what gives them the high melting points, and that's what makes them a solid. So most monosaccharides and disaccharides are sweet tasting, they're digestible, and they're non-toxic. So not all of them, but most. So glyceraldehyde and fructose are not that way. So they can form, looking at the wrong thing. So glyceraldehyde and fructose are not as sweet tasting, fructose is, but digestible and non-toxic. So we're going to look at some important monosaccharides. So one you're going to see a lot of is glucose. So glucose is an aldohexose, which means it has an aldehyde. So on our structure here, here's our aldehyde, and then it has 6 carbons. So 1, 2, 3, 4, 5, 6. And it is a product of photosynthesis, and it's found in fruits and veggies and corn syrup and honey. It's stored in starch and glycogen, and it's a building block for some disaccharides and polysaccharides, and we're going to see that a little bit later when we look at those. And glucose level in blood is regulated by the hormones insulin and glucagon. Okay, galactose is an aldohexose, which the only difference from glucose is this hydroxyl group right here. So in glucose, it's over here. So that's the only difference between the two. So we have the aldehyde up here. We have the 6 carbons in the chain. So just that one hydroxyl group in a different position makes it into galactose instead of glucose. So it is a component of brain and nervous system tissues, and it's found in plant gums, pectins, and milk as half of the disaccharide, so lactose. It is converted to glucose in the metabolic pathway. And the next one we have is fructose, and it's in ketohexose, so there's a ketone group in there. So it's right here. And since it's a hexose, there's 3 carbons in the chain. 1, 2, 3, 4, 5, 6. So it's found in fruit and honey. It's a component of sucrose, and it's produced by hydrolysis of cornstarch to make high fructose corn syrup, and it's sweeter than sucrose. So it's used to sweeten drinks and prepared foods. Then we have ribose. So it's an aldopentose. So you can see up here is our aldehyde. And then we have 5 carbons, 1, 2, 3, 4, 5. And it is a part of ribonucleic acid, and it's involved in protein synthesis. And then the last monosaccharide we're going to look at is 2-deoxyribose, and it's an aldopentose. So the only difference between ribose and 2-deoxyribose is here on ribose we would have the hydroxyl group, but instead we have just a hydrogen. So anywhere there's a blank, those are just hydrogens. So that's what makes it different from ribose. So, and again, it's part of DNA. Okay, so now we're going to look at reactions of monosaccharides. So the first one we already know about. It's a reaction with oxidizing agents. So we know previously that when an aldehyde is oxidized, so here's an aldehyde, when it's oxidized, it produces a carboxylic acid. Oh, not the H. OH. And so that's what we're going to see happening in our monosaccharide. So when the open chain aldehyde is oxidized, it displaces the equilibrium that was established between the open chain and the cyclic structure. So you can see right here the equilibrium symbol, right? So we have the open chain right here. We have the cyclic structure right here. So that's an equilibrium until the open chain is oxidized. Okay, so once that happens, the equilibrium is displaced and the open chain is oxidized, and we are forming the carboxylic acid, but really the carboxylate ion here. So carbohydrates react with oxidizing agents, and if they do, they're classified as reducing sugars. So that's basically what we did in the lab when we used Benedict's reagent, and it said if we had brown, orange, or red precipitate, then that was an indicator that there was a positive test for the presence of a reducing sugar. So all of them, I think, I'm trying to remember exactly what we had, but water and starch. So everything else was a positive test except for water and starch. I think starch was the right one. So that's what's happening. The open chain of the monosaccharide is being oxidized to get the carboxylate ion. So we saw that even in the ketones. So this is an aldehyde. We saw that with ketones as well. So we know that when we, normally when we try to oxidize a ketone, that doesn't happen, right? You can't oxidize it. But if a ketose is in a basic solution, then they are reducing sugars. So what happens is, here's our ketose. All right, we've got that ketone group there. And then when it's put into a basic solution, so OH, that represents our basic solution, then it undergoes a rearrangement of the atoms. So first, our double bond that's on the ketone moves to be in between the carbons there, and now hydrogen is added to that oxygen. So now we have a double bond, and we have a hydroxyl group. We actually have two hydroxyl groups. Okay, so it's called an enediol because ene for the double bond and diol for the two hydroxyl groups. Okay, then what happens is the double bond moves again. It rearranges now to form the double bond here on the end. So now we have an aldehyde, or an aldose, and that can be oxidized now to the carboxylate ion. So even ketoses are able to be oxidized if they are in a basic solution because of that rearrangement of the ions, or of the atoms. So remember, the enediol is formed, and then the aldose is formed, and then it goes to the carboxylate ion. So it is a reducing sugar. Okay, monosaccharides can easily be reduced to produce sugar alcohols. So we just looked at oxidation. Now we're going to look at reduction. And we can go from a monosaccharide to a sugar alcohol. And we call these alditols. So the sugar is exposed to hydrogen in the presence of a catalyst. And then the sugar alcohols are named by changing the OSE in the monosaccharide name and ending in the ITOL. So glucose becomes glucodol. Okay, so here's an example of that. So here we have hydrogen. Whoops. Let's try that again. We have hydrogen. We have our catalyst here. So this is glucose, but when I react it with hydrogen in the presence of the catalyst, then my aldehyde here goes away. So basically I'm adding 2 hydrogens. So I get a hydrogen attached to carbon and a hydrogen attached to oxygen, and my double bond goes away. So this is called a sugar alcohol. So these sugars are used as sweeteners in many sugar-free products. Okay, next we're going to look at glycoside and disaccharide formation. So hemiacetols react with alcohols to form an acetal. So we saw that when we were looking at aldehydes and ketones. So we know that if a hemiacetal reacts with the alcohol, the alcohol part attaches, and then we have 2 OR groups bonded to the same carbon. That makes the acetal. So the same thing happens with a monosaccharide. So when a monosaccharide reacts with an alcohol, it's going to produce an acetal. So, but here, the hemiacetal is in the form of the cyclic structure. So the hemiacetal is right here. Okay, so we have the OR. Well, let me draw this a little better. So here's the carbon, right? Here's the carbon, and then we have the OR group right here, and then the OH group here. So that makes the hemiacetal. Okay, so when we react the hemiacetal with an alcohol, we form the acetal. So now this carbon has 2 OR groups attached to it. So this right here, this red bond right here, is called a glycosidic bond, and it can be either alpha or beta. So I'm going to show you on this next slide. Okay, so that was just the monosaccharide reacting with an alcohol, but now we're going to look at that glycosidic bond forming between 2 monosaccharides to form a disaccharide. Okay, so here again we have the hemiacetal, and then we have an OH group from the other monosaccharide. So we get the same reaction taking place, and here again is that glycosidic bond. Okay, so this one is an alpha glycosidic bond. So if it's pointing down, if the bond is pointing down, then it's considered an alpha. And if it's pointing, let me write that down. So if it's down, it's going to be an alpha glycosidic bond. If it's pointing up, then it'll be a beta glycosidic bond. So those are the 2 things that can happen there. We can either get the acetal, or we can form a disaccharide. And those monosaccharides are connected with the glycosidic bond. Okay, the reverse can also happen. We can, if hydrolysis takes place, then it's going to break apart the monosaccharides. So I should say it'll break apart the disaccharide and produce the monosaccharides. So this takes place during digestion of carbohydrates. Hydrolysis does. Okay, the last reaction we're looking at with monosaccharides is the formation of a phosphate ester of alcohols. So it can be formed when a phosphate group bonds to the oxygen atom of an OH group. So you can see that here we have glucose, and here's our OH group. So now the hydrogen goes away, and the phosphate is able to bond with the oxygen there. So it's able to do this. The phosphate is added from ATP. So you can see right here, we talked a little bit about ATP and ADP previously in the carboxylic acid chapter. So that's where the phosphate's coming from.